Hello, thanks for joining us for this presentation of using the SAWGraph to query information about PFAS contamination and PFAS transport. This is part of the SAWGraph project, the Safe Agriculture Products and Water Graph, which is a Proto-OKN project focused on contamination in the nation's food and water system. I'm Torsten Hahmann, Associate Professor of Spatial Computing at the University of Maine, and leading this project, and will give you a short introduction and then hand it over to some of our team members to show you what is possible with SAWGraph right now. So at the back end, SAWGraph consists of 6 interconnected knowledge graphs that kind of organize information about PFAS contamination, but also supporting information about facilities and their industries, hydrological features such as surface water and subsurface water, as well as a spatial graph that kind of connects these features. So in addition to what we have here, we have a 6th graph that covers crops and soil information about those locations and the different administrative regions. So what kind of use cases does SAWGraph support? We have organized the types of questions that we identified as relevant to stakeholders from government agencies into 3 use cases. The first one is about analyzing the existing test results, like sample results, like finding what wells are near certain locations, like where we've seen or detected PFAS contamination or contamination by specific types of PFAS. Or the other way around, what samples are near certain types of facilities, for example, near landfills or near plastic manufacturing facilities. The second use case is about assessing the impacts on different water and food supplies. For example, how are drinking water supplies impacted? Are gravel wells more impacted than bedrock wells? Or what kind of crops are growing near places where high PFAS concentrations have been detected? The third use case focuses on tracing PFAS, like through the primarily surface water, but also subsurface water system. So the first question is about potential subsurface transport via aquifers, asking about what water wells are potentially drawing from aquifers connected to other wells where we've already detected high PFAS levels. But we can also trace through the stream network, like the surface water stream network, to identify either facilities that are upstream of concerning sample results and even grouping them together. What kind of industry sectors do we see upstream there that might be the cause of the PFAS contamination? Or the other way around, like if you want to understand a particular type of industry or type of facility, whether it might contribute to PFAS contamination of water sources, water resources, you can look at samples downstream from a particular facilities and kind of look at what kind of substances have been detected there, or what kind of concentration do we find, or do we even have enough samples there. So we will give you some examples of what is possible, how to answer some of these questions. The first use case we will go through is the first one, the questions about testing, but then we will continue with some tracing examples before finishing up the presentation with 2 examples of assessing impact, the two questions here. Let me hand over to Katrina Schweikert.
Thanks. I'm Katrina Schweikert, a PhD student in the Skylab at the University of Maine in the School of Computing and Information Science. So I'm going to go through an example that looks at the first use case, which is related to testing. So where has testing happened, or where should we do further testing? And what are the test results showing? The specific query that we're going to look at is what samples are near certain types of facilities. And we can adjust the type of facilities that we're concentrating on. We'll take a look first at sewage treatment facilities, which are one site of accumulation of PFAS due to it filtering through the water system and into the sewage treatment facilities from both industrial sources and also just households. So in order to answer this question, we use data from two main graphs here. So the facilities and industry graph, we want to start by finding facilities based on their industry. And then connecting through the spatial information, so particularly this grid of spatial information that we have in S2 cells, we then find observations, so specific samples that were taken near those facilities. So this is really a spatial question. So ultimately, we would like to produce a sort of map visual. And so we've broken this question down into kind of three sub-questions that give us some different spatial information. First, we'll take a look at running this on the Frink server. So the first thing that we want to do is find facilities of a particular type. So we're running a query here to find facilities. And the key thing here is based on their industry code. And this one is the North American Industry Classification Scheme code for sewage treatment facilities. And then we're just getting their location, so their geometry, so that we can map it, and some additional information about them. And this is going to take a minute to run as we have data for various countries or various states already loaded into our graph here. So right now we have 1,900 results around the country of sewage treatment facilities. Once we find those facilities, we then want to look around them, so find the neighborhood. And this is where we leverage the spatial graph, so information from this grid of S2 cells that we have and S2 cells that are touching the ones that specifically contain the facilities. And this gives us our sort of neighborhood that we're looking for to find samples within. So we can run this one as well. You already saw the result there, but this one takes a minute as this is, again, capturing quite a lot of data around the various states that we have loaded into our graph. And then the third step here, lastly, would be to use that neighborhood to then find our sample points. So we've simplified this a little, just looking at some specific S2 cells that I've just listed out here. We can do this more dynamically by connecting these queries together as well, but just to simplify what we're looking at. So this one is looking for observations and getting some information about those observations and the sample results that were found there. And this should give us, each location, what is the maximum concentration that was found there, which substances were found there. If we expand this, you can see the specific list of PFAS individual chemicals that were tested for samples taken at that site, what type of material was tested, and also the location so that we can map it. We've also used these queries in a Jupyter notebook to demonstrate how to do the mapping. So this runs the same set of Sparkle queries through Python. And we'll see if we can get this running. So we looked at sewage treatment facilities, but again, we could quickly switch out which type of facility we want to use using a different NICE code here. Here's some of the ones that are commonly looked at for their usage of PFAS or their suspected usage of PFAS. And we break the queries again down into facilities and their locations. So here's the same Sparkle query. The neighborhood around the facilities, similar to what I demonstrated, and then the sample, details about the samples taken in the neighborhood. We need to just do some quick prepping of that data for mapping, and then finally we can produce a map result of that information. And these also have some pop-ups so we can look at the specific chemicals that were tested there, just like we saw in our table result, the maximum concentration, the count of test results, etc., based on all of these locations near sewage treatment facilities. This is also saved as a file, and some of these examples we have hosted on our website, but we can look at the specific map standalone, and here's a second example. This one looks at paper manufacturing facilities, and they are divided into many of the different specific industry codes under the general industry group that is suspected of utilizing PFAS. So that was one quick example of a query that we can run using the primary use case of where has testing occurred or where should we do testing. Now we'll take a look at some tracing, and I will hand it over to David Kedrowski.
Hi, I'm David Kedrowski. I'm a PhD student at the University of Maine in spatial computing. And I'm going to take a look at 3 different queries today. The first 2 are kind of almost like mirrors of each other. We're going to take a look at facilities that are upstream of existing sample points. And then a second query, we're going to flip that around and look at sample points that are downstream from specific facilities. And we'll see that in practice, they are 2 kind of very different queries and give us 2 different ways of looking at PFAS contamination, even though technically under the hood they are very similar. We use the PFAS graph to get to the sample information, the SAWGraph to get to facilities, and hydrology to do the tracing, and then spatial we use to pretty much kind of connect everything together. We also use it to kind of scope the sample points in the first query and the facility in the second query, and I'll show how all that works when we get to a map here in a moment. The third graph, or the third query, excuse me, we're going to take a look at groundwater tracing. We're going to find a specific well that has shown signs of PFAS contamination, and then find an aquifer that it sits above and most likely taps into, and then take a look for other wells that tap into the same aquifer, assuming that the PFAS could be spreading through that aquifer from the contaminated well. So it gives kind of a sense of other places to test. So a lot of these queries are kind of one use case, but they tend to cover multiple use cases. All right, so the first query, what facilities are upstream from specific types of samples with high PFAS concentrations? We've designed this query in a Jupyter notebook so that you can choose different types of PFAS. In this case, I'm going to be looking at PFOA. We can also look at different material types. So for this is groundwater, but we could be looking at drinking water, wastewater. We have a variety of different material types that could have been tested. I'm going to be scoping to a specific county in Maine. So I'm only going to be looking for sample points that are within Cumberland County, Maine. And then we can assign some minimum and maximum concentration values that we're looking for as well. This makes use of a total of 3 different queries. The first query is looking for those sample points within Cumberland County, and that's the region code that you might be able to read there. It then finds S2 cells that each of the sample points are located in, and then looks for essentially rivers or streams that are near using those S2 cells. And then it also pulls up a bunch of information just specific to the sample in terms of the results that were found from testing. The S2 cells are transferred into a second query where we start with the what would be downstream flow paths of rivers, and then we trace upwards and try to find all of the streams and network pieces that are upstream from the downstream pieces, and therefore from the sample points. And then we take S2 cells that are along all of those and we transfer them to a third query. And in that third query, we look for facilities that are within those S2 cells along those rivers. So we're finding facilities that are near and along the rivers. We end up with a map that looks like this. And what you see right now is Cumberland County, Maine. So the query made sure it only found samples within that county. And the size of the circles has to do with the relative size of the maximum value found at these different locations. Once we have these sample points, then we can start to take a look at the flow lines that emanate upstream from them. And as you'll see here, they actually go quite a ways. So we can start to look at the impact within Cumberland County outside of it. And once we know those flow lines, then we can look for our facilities along the way. And we have this set up so you can look at different facilities. So right now, this is fabricated metal product manufacturing. We could look at paper manufacturing. Those are a little hard to see, but they're there in blue. We could look at specifically chemical manufacturing. So you could look at different sources potentially of PFAS along the way here as well. So we're looking at samples downstream from specific types of facilities. So you can choose a lot of the same information as for the previous query, but I've added in the ability to choose a specific industry for those facilities. This is pretty much all run from a single query, but it's going through and doing something very similar. And I'll kind of talk through exactly what all this does in the map. I think it's a little bit clearer to see there. And the couple of extra queries as I'm scrolling through there, just to pull up pieces of geometries for the map itself. So here I'm looking at Kennebec County in Maine, and what we're going to do is pull up facilities, and these facilities, I should have mentioned this back up here, are specifically in the wastewater treatment and disposal industry. So these are the facilities we have within Kennebec County for those. And we do flow lines again, but we're looking downstream at the flow line. So instead of getting this huge network upstream, all the different possible places things could be coming to get to a downstream point, we're looking only downstream. So it's a much simpler looking set of flow lines that we end up with as a result. And then we can look at samples along the way. And as for both the previous query example as this one as well, you can point and get pop-up menus that have additional information, in this case about sample results, or we could look at the facilities and find out more there, and we can add other information from our graph. We can even look up some information about the flow lines themselves, although they're a little bit difficult to point at, but there's a little bit about a flow line that we're seeing there. The third example I have is the private well example. In this case, not quite as many attributes to choose from, but have a couple of different sample points to choose from. In this case, it has this arcane value of 72822. I'm going to be looking at Kennebec County, Maine, looking at groundwater samples, since aquifers and wells are all about groundwater. This query also pretty much does everything it needs to within a single query. And we end up with a map that looks something like this. The orange point here in the center, oops, sorry about that, is our sample point, and it's a single sample point. There's some information about it. This is the aquifer that it is above, so we're assuming that there's some connectivity here. One of the things that's kind of interesting, I think, about this query is that when we look for the wells that are also associated with this aquifer, we get wells not only that show above the aquifer on the map, but we also get wells that are nearby and outside. And that's one of kind of the artifacts of the fact that we use S2 cells to determine geometries and this idea of, or the geographies and the closeness, the spatial relations between things. If I turn on the S2 cells that intersect with the aquifer, we can see they create a bit of a larger area than just the aquifer itself, and that's why we're picking up more wells. We actually view this as potentially a good thing because it shows us not only the very specific wells that are in the aquifer, but it's also some very nearby wells, which may be, and actually likely are connected to this aquifer and to each other through this system as well. Kendall's going to come up next and talk to you about some analysis work.
Hi, I'm Kendall Phillips. I'm a master's student in the Spatial Information Science program at the University of Maine. And I've been using SAWGraph to kind of assess some impacts and look at gravel wells and bedrock wells in Maine. So typically gravel wells are much shallower than their bedrock counterparts. So the thought is they should have higher levels of contamination since the PFAS has less distance to travel in order to contaminate these wells. So what I did was associate the sample point locations with well locations through the spatial graph. So when the sample points were taken, it's not recorded what type of well they were taken from. So I used the SAWGraph graphs to kind of associate those values with each other. So in order to figure out if a sample point was likely from a bedrock well, I'd look to see, is it near a bedrock well? Is it near a gravel well? What sort of well it was near. So the first part was to query to find both detects, which was anything above 4 parts per trillion, and then non-detects, which is anything below 4 parts per trillion, for sample points that are in the same S2 neighborhood as gravel wells. So that would be the S2 neighborhood, or the S2 cell that contains the gravel well, and then the 8 surrounding cells. So that queried over the PFAS, spatial, and hydrology graphs. The chemicals that this query focused on were 2 of the more common ones, PFAS and PFOA. And then the detected sample points were, the outliers were filtered out to just kind of avoid any errors with taking in the data. So any numbers that were like extremely huge were filtered out of the results. And then non-detect sample points were also found, and they were assigned a random value between 0 and 4. And so then the second part of this query, well, the second query was to find both detect and non-detect sample points that are near bedrock wells. So there, I just looked at an S2 cell containing at least 1 bedrock well, but no gravel wells. And for this query, I kind of casted a smaller net because there's way more bedrock wells in Maine than there are gravel wells. So if we did the S2 neighborhood, we would get virtually every sample point in the state. So that's why I just kept this one to a smaller area. So where there was a bedrock well, but no gravel wells. And then I also removed any samples that were a result of the previous query, so that are potentially gravel samples. So then nothing was double counted. And then this queried over the same 3 graphs, PFAS, spatial, and hydrology, and then focused on those same 2 chemicals. Next slide. So after analyzing that data, I was able to create a box and whisker plot that kind of shows the distribution of results. And as you can see, as far as the 25th and even 50th percentile, there's not much difference between bedrock and gravel wells. But once you head up towards the 75th percentile, you start seeing a difference. You have a far greater number of gravel samples that had much higher numbers, which is what we kind of expected to see. Next slide. So then here are 2 tables that just kind of summarize what the previous plot shows. So on the left, there's a summary of well samples with detected quantities that are likely from gravel wells. And on the right, you have the same thing, but with bedrock, potentially bedrock well samples. So you have the count. There were about 1,500 gravel samples with detected values and about 800 bedrock samples with detected values. And then, as you can see, the mean and standard deviation vary greatly between bedrock and gravel. You have much different concentrations for that. As far as just like in the previous plot, the 25th and 50th percentile do not vary much between the 2, but then as we move up towards 75 and the max, you have very different concentration levels for those 2. So this shows that kind of mimics the pattern we're thinking to see. We've got higher concentrations in gravel wells that are typically shallower than we have in bedrock wells that are deeper. So I will now hand it over to Adrita.
Hi, I'm Adrita. I'm a PhD student in the Department of Computer Science at Kansas State University working with AC Lab. I'm going to talk about some of the impacts of PFAS contaminants on crop plants. Here you are seeing a standard query. We have run this on GraphDB, and you can see that it's about finding all the F2 cells in a particular town. We have put Auburn in one of the counties, Maine, and that grows root vegetables that have a particular PFAS contamination detected in the soil samples at a certain level. For here, for this we have taken 4 nanogram per liter or above. Through this query, you could see what are the if you want to see a particular crop that's growing near near the sample points that also have PFAS contamination detected for it. For this query, we are mainly using 3 interconnected graphs, which is spatial, PFAS, and crops and soil graph. In the spatial graph, we have all the spatial informations about different locations. And in the PFAS graph, we have the sample points and different kinds of sample materials that they are detected from, and also the related PFAS related information and the chemical information on that graph. And in our crops and soil graph, we have structured crops data from NOAA, where you have different crop types which are associated with different locations, and also we have mapped them to some categories and subcategories, so you can broadly query the graph to find certain type of crops like grains, or in this case we are looking for root vegetables which might have PFAS intake through soil samples that are detected near them. Next slide, please. Okay, in this slide, I'm going to go a little bit into the specifications of the query that we have, and this is a template you can use to query the graph and find different kind of information from here. In the first part, we have defined the specific location. Here, the DCGO ID would specify the location that you want to query about. And in the PFAS graph, if you go over, you can find different sample points that is associated with that particular location. And then you go down, you can define the particular sample categories. For this case, we have specified it to only to the soil samples that are found in that location. And you can also change the values of the PFAS contaminant elements that have been found. And then you can change those as well to your desired specifications. Next slide, please. And in the last part of the query, you can query the crops and soil graph to find specific information of different type of crops. Here, you could find the different observations associated with each S2 cells, and then here you can also specify the time where if we have specified for the year 2021. And then for that observation on that year, you can find what are the crop categories that are grown near that area. Here we have defined it to root or tuber vegetable. You can change the subcategory to leafy vegetable or grain or anything that you want to do. You can also specify to a particular crop type as well, which you have to you can specify all those information to find desired outputs. And in the next slide, I'll show the results of this query. Here you could see different locations the locations there was the soil samples were found with the PFAS contamination with 4 nanogram per liter or above. And in the second column, you could see the PFAS contamination levels that are associated with those S2 cells that is aggregated all the sample points found in that particular S2 cell. And also you could see in the third column that those areas have crop growing lands, which are root or tuber vegetable, which are growing on those lands near those S2 cells and the sample points. So here with this query, you could see the impacts of PFAS contaminations found from different sample types, and also which has locations near those sample points which are growing different kind of crops. And through this query, you could find all those information. That's all from me.
So this is just an example of some of the kind of queries the graph currently supports. We're still growing the graph both geographically, but also the type of samples we ingest and the kind of questions we demonstrate, and we will share updated information and examples on our website. But this would not be possible without everyone on the team. So thanks to the entire team for making this possible, and also thanks to the funders, to the Proto-OKN initiative, of which SAWGraph is a part of, as well as USDA's funding through the Humane ARS Center of Excellence. And of course, all the government partners and collaborators for providing input that define the use cases and the example questions, and helping us kind of explore what is possible, and how we can use the data to actually help decision making and analysis. If you have any questions, please reach out. We're happy to answer any question by email or set up a meeting. So we welcome questions anytime. Thank you.